what are your current position and field of interest? Could you please describe the group you lead, its facilities and technologies? What are the main objectives of the group, and what kind of research activities are currently ongoing? What are the perspectives for future research in the biomolecular field over the next years? And how might artificial intelligence impact research? What advances are expected, and how could they contribute to the development of new technologies and research approaches in this field? And now we move to Professor Umberto Malapelle. Umberto, up to you.
Hi everyone, my name is Umberto Malapelle. I'm the chair of Predictive Molecular Pathology Lab at the Department of Public Health in University Federico II of Naples in south part of Italy. And I'm an associate professor at the same department at the same institution. And our history as a group starts with the KRAS analysis in colorectal cancer to define the treatment decision-making for the oncologist. And we founded this lab really focused on the management of different type of cytohistological samples. And in the first part of our history, we are really focused on the management of cytological samples and the other biofluids. And this was really helping for us to move this knowledge also in the field of liquid biopsy. And our current research interest is really in the field of liquid biopsy and the other biofluids to assess different type of biomarkers or predictive biomarkers for different type of target treatment in solid tumors, like as non-small cell lung cancer, metastatic colorectal cancer, melanoma, gastrointestinal stromal tumors, and so on. In all the solid tumors in which we need to analyze predictive biomarkers to define the therapeutical strategies for our patients. Currently, I am also the scientific secretary of International Society of Liquid Biopsy, and we work also on different type of educational projects to increase the awareness about the use of liquid biopsy in clinical setting, and we are trying to bridge the gap between translational research and clinical setting in the use of liquid biopsy, because if you think that in Europe we have more or less 43% of patients that are not able to have a complete status defined for biomarkers to evaluate the first line of treatment in a small cell lung cancer setting, and we are trying to bridge this gap to combine the data coming from liquid biopsy and tissue. And now we are really convinced that the future probably is in the integration of the different type of biometrics that you have. An example, by combining data coming from liquid biopsy and tissue to fully exploit the possibility to have a morphomolecular evaluation for our patients. And we are working on 3 different projects in non-small cell lung cancer and colorectal cancer to try to identify the role of a private set of alteration in liquid biopsy or in tissue regarding the treatment performance for these patients when we are able to find, in example, one mutation in blood and we are not able to find this mutation in tissue and vice versa. And then we are working on the possibility to integrate different type of biological level in liquid biopsy, not only at DNA level, but also RNA and proteomic levels, also to take into account other type of strategies like as fragmentomics in liquid biopsy, and we are trying to combine the information coming from fragmentomics, mutation, and RNA in liquid biopsy and tissue for these patients. And I think that this is a really exciting era because we have a lot of methodologies. We are working with the different type of tools that we have 10 years ago, in example. In our lab, we have the access to different type and different generation of next generation sequencing, but also other type of next generation methodologies like as multiplex color code, barcode technologies for gene expression analysis and other type of analysis. And it's really nice to have the opportunity to combine the data coming from this type of methodologies on different type of biometrics, tissue and liquids. And I think that the future is in the marriage among all this type of information that we can have. And in this view, is absolutely needed the help of other type of intelligence, like as artificial intelligence, because we need to combine simultaneously thousands and thousands of data coming from different type of analysis. In example, in our lab, each year we have more than 20 tera coming from only NGS and the other 40 tera coming from gene profile, and it's really complicated to storage this data, to combine this data, to interrogate simultaneously this data. And it's obviously that in future the role of artificial intelligence in this landscape will be more and more important. And so on the other hand, we are working on to try to find and fine-tuning biomarkers for immunotreatment in gynecological cancer, like as endometrial cancer and ovarian cancer, because we start works 5 years ago with homologous recombination disease assessment in this type of cancer, and now we are trying to figure out the less predictive value that we have with some type of alteration, and we are trying to combine gene profiling and mutation assessment on the same type of patients to verify if we are able to make in place and a fine-tuning to better define the responsive patients among all the patients that we are analyzing. And the last thing is that I am really proud to be part of this group and to have the opportunity to discuss with all of you this type of things. Thank you.
Thank you very much, Umberto. You and Giuseppe mentioned several times the liquid biopsy. What is the state of the art in terms of standardization and costs? As I think that these are two pivotal points for the extensive application of the liquid biopsy.
Fully agree. Great question. The first one regarding the application in clinical practice today is in non-small cell lung cancer patients in the advanced stage. When the tissue is not enough, we have the ability to proceed with the analysis of circulating tumor DNA for all the clinical relevant biomarkers approved, like as EGFR, BRAF, KRAS, ALK, ROS1, RET, and 3K, and so on. Another application is in the landscape of breast cancer to analyze PI3 kinase mutation. And now another upcoming application will be in the analysis of ACR1 alteration in the context of breast cancer patients. And outside the circulating tumor DNA, and a clinical application is in the enumeration of CTC in breast cancer to define the prognosis of patients. This is not approved in Italy, but in other countries of Europe is approved. And regarding the state of the art, if we see an example, the last statement paper coming from International Association for the Study of Lung Cancer, after that, and a multidisciplinary tumor board decide that is needed, you have the ability to proceed with a complementary approach starting in basal setting on tissue and liquid biopsy for non-small cell lung cancer patients. This is the state of the art. Regarding the cost, we need to pay attention that if we are thinking, an example, by the using of next generation sequencing on liquid biopsy, we need to have a higher coverage respect to the coverage that we have for tissue-based analysis. And this means that you need to run a smaller number of samples for each run. And this is resulting in the application of the cost for a liquid biopsy in reagents view respect to any tissue-based analysis. This is true, but pay attention that now we have a really escalation of the methodologies, because when we start to apply liquid biopsy 5 years ago, the cost is 3 times the cost that we have today in place for the liquid biopsies analysis. And in future, probably we will have the opportunity to apply liquid biopsy also in metastatic colorectal cancer after a first line of treatment, before to decide if we are able to proceed with a very challenging treatment with a drug that we used in first line by monitoring the mutant allele fraction of KRAS and NRAS alteration, in example for metastatic colorectal cancer. And another application in future, I'm absolutely convinced that will be the monitor minimal residual disease by using liquid biopsy, not only by using NGS, but also, an example, more accessible technologies like as digital PCR.